All man-made electronic machines use a standard source of electricity, moving charged particles like those shown here. But in cells, there are no plugs or wall outlets. Instead, cells use a molecular power plant called the F1F0 ATPase to make molecules of ATP, the standard source of cellular energy. Let's now look closely at this magnificent machine. This massive molecular motor has close to 40,000 atoms and can make nearly 100 ATPs per second. Each cell contains thousands of ATPases, each of which operate at nearly 100% efficiency, a level not even approached by any man-made motor. Its design is so elegant and optimized that species as different as bacteria and humans have nearly identical ATPases, whose structures have remained relatively unchanged throughout millions of years of evolution. Anchored in the inner mitochondrial membrane, the ATPase is divided into two main domains, the F0 domain embedded in the membrane and the adjoining F1 domain shown here above the membrane. We will focus first on the F1 domain, the site of ATP synthesis. There are 3 alpha subunits shown in green and 3 beta subunits shown in blue. These surround a rotating central stalk composed of the gamma, delta, and epsilon subunits. The peripheral stalk, composed of B and D subunits, extends from the membrane to the top of the F1 domain, effectively anchoring the alpha and beta subunits so that they do not rotate with the central stalk. Interactions with the rotating central stalk lead to alpha and beta subunit conformational changes, which we can see here from the ribbon representation of a beta subunit. For each complete rotation of the central stalk, the alpha and beta subunits undergo 3 dramatic conformational changes, each corresponding to a particular step in the ATP synthesis reaction that converts ADP and inorganic phosphate into ATP. To explore these reaction steps in greater detail, we are entering one of the 3 ATPase active sites, which are at the interfaces of alpha and beta subunits. ADP only enters the active site when the alpha and beta subunit atoms are in a particular orientation that favors its association. A host of hydrogen bonds among beta chain amino acids and water molecules stabilize the adenine substructure within the active site. Ribose similarly interacts with ATPase atoms via hydrogen bonding. Diphosphate coordination in the active site involves several hydrogen bonds, as well as salt bridge interactions that require a magnesium divalent cation. Once ADP and inorganic phosphate are appropriately positioned in the active site, a transition state is formed in which the ordinarily pyramidal geometry of inorganic phosphate becomes planar. Upon resolution of this planar transition state, inorganic phosphate bonds covalently with ADP, yielding an ATP molecule. The subsequent conformational change of the ATPase breaks the hydrogen bonds stabilizing ATP, leading to its dissociation from the active site. This cycle of association, synthesis, and dissociation repeats very rapidly. We have just seen that conformational changes caused by central stalk rotation drive ATP synthesis. But what causes central stalk rotation in the first place? The answer, namely the F0 domain, lies in the membrane and involves the regulated flow of protons. The F0 domain consists of a ring of identical transmembrane C subunits. Next to the ring is the A subunit, for which no atomic level structure is known, hence its depiction here as a faded shape. There are 2 other key players in the function of the F0 domain, the membrane, shown in gray, and protons, shown in yellow. On this side of the membrane, protons are relatively scarce, in stark contrast to the intermembrane space where they are abundant. As we'll see shortly, this electrochemical proton gradient across the membrane provides the energy for ATP synthesis. Each C subunit has a proton binding site at an aspartic acid residue shown here in orange, and the current model for F0 domain rotation suggests that the A subunit regulates proton binding. Specifically, it is thought that of the 4 subunits in association with the A subunit, the 2 innermost are never bound to protons, and the 2 outermost are the only positions in the ring where proton exchange occurs. If we assume that only protons above the membrane can interact with the leftmost subunit, while protons below only interact with the rightmost one, then the mechanism for rotation becomes clear. Because of the proton gradient, the rightmost subunit is proton-bound frequently, whereas the leftmost subunit is rarely proton-bound. In this condition, rightward turns of the C ring, driven by Brownian motion, do not violate the A subunit enforced rules, since the middle 2 positions still lack bound protons even after the ring turns. In summary, the proton gradient, the binding rules imposed by the A subunit, and rotation driven by thermal fluctuations cause rotation of the F0 ring and consequently the F1 central stalk, thereby providing the energy to synthesize new ATP molecules. In this animation, we have learned about the F1-F0 ATPase, a highly conserved cellular power plant present in organisms ranging from bacteria to humans. The membrane-embedded F0 domain uses an electrochemical gradient to rotate the F1 domain's central stalk, causing conformational changes in alpha and beta subunits that convert ADP and inorganic phosphate to ATP.